Hi, Michelle Glass here. Welcome to our first episode of Chapter 10, Lecture 10 series. Our topic in Chapter 10 is all about the DNA molecule, deoxyribose nucleic acid. And we're going to begin with our first video talking about how this was the molecule, how it was discovered that DNA was in fact a hereditary molecule. So it wasn't until 1952 that Martha Chase and Alfred Hershey were able to successfully determine that the hereditary molecule was in fact DNA. Prior to that, the discussion was centered more around proteins as the possible hereditary molecule because of the complex shape of proteins. Hershey and Chase are famous for their research that they did using a bacteriophage named T2. Now, a bacteriophage in general is a virus that infects bacteria. And we know viruses now are consisting of protein capsules, and then the nucleic acid is the molecule inside. And at the time, the question was, what component is actually the hereditary molecule? Is it the protein or is it the nucleic acid? Now, viruses are going to inject the hereditary molecule into the host cell and use the host cell in order to replicate. So Hershey and Chase decided that they would tag proteins and tag DNA and see which molecule was actually injected into the E. coli in order to produce more virus. So here we're seeing a bacteriophage injecting its hereditary molecule into the E. coli, and as a result of that, producing more bacteriophage. And so the question is, okay, what is the hereditary molecule? Is it a protein or is it nucleic acid? So what they did, their basic design was to take both E. coli and T2 in a flask, blend it, and then separate out the components. And you're going to see the phages and the fluid at the top, and you're going to see the E. coli pellet at the bottom. And the question will be looking at whether we've tagged proteins. So in one tube, they're going to tag proteins, and in another tube, they're going to tag nucleic acids. And then they're going to look to see what's actually down there in that pellet with the E. coli. So let's talk a little bit more details here. So again, our question is basically, is protein the hereditary molecule or is it DNA? So to begin, in one experiment, Hershey and Chase took E. coli T2 and radioactive sulfur and put it together in the flask. Now the radioactive sulfur is going to get incorporated into the protein structure. So in this first example, the protein will be radioactive. In the second set, they did E. coli and T2 with the radioactive phosphorus. Phosphorus is going to be part of the DNA structure. The T2 in each case is going to infect the E. coli, and then we're going to see in the flask with the radioactive sulfur that the sulfur remains in the protein solution rather than in the cell pellet. Whereas in the radioactive phosphorus flask, the radioactivity is with the DNA molecule, and that's going to be in the cell pellet. So the fact that the DNA is the part that's fluorescent and it's down in the cell pellet tells us that that is the molecule that was actually injected into the E. coli. So again, because the cell pellet with the E. coli is the radioactive part at the end of the experiment, that tells us, and that told Hershey and Chase, that the T2 bacteriophage injected the DNA molecule into the E. coli in order to replicate. So our hereditary molecule is DNA molecule. So since 1952, we've come a long way in our understanding of genetics and heredity, and we're going to continue that discussion as we move forward. So stay tuned.